ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay akyat. Hawak kamay tayo'y aangat, lilipad. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas. Maayos na bukas ating iparanas sa bayang Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ang aming hatid. Tara na, kaibigan, huwag kang magpaiwan.
Mabuhay at magandang araw sa inyong lahat. Welcome to the second day of the 9th installment of Virology and Vaccine Institute of the Philippines, or VIP program webinar series. I'm Joseph Ancla from DOST ITDI VIP program, and I will be your host for today. This two-day webinar is part of the Virology and Vaccine Institute of the Philippines, or VIP's information and education campaign on virology and other related topics. This learning session is made possible by the collaborative efforts of DOST Central Office, DOST ITDI, DOST PCHRD, DOST Picard, and DOST Balik Scientist Program. So before anything else, let's check first the number of participants here in our Zoom meeting room, as well as our audiences from our Facebook and YouTube live streams. So allow me to check. As of now, we have participants. We have 101 participants from Zoom, and I can see that some of our participants are from LPU. So we have from Lyceum of the Philippines University. Meron din po tayo from MSU, CNSM. Meron din po tayo sa PLM, sa Pamantasan ng Lungsod ng Maynila. Meron din po tayong participants from PRMSU. So we also have participants from Rescue 922 and South SEED LPDH. We also have participants from TSU and University of the Philippines, Manila and Los Baños. We also have participants from VSU. So thank you very much for attending our seminar this afternoon. Moving on to our Facebook Live, we have 28 viewers, and for our YouTube live stream, we have 9 viewers. So before we begin the second part of our lecture, let us first watch some of the video clips about Virology and Vaccine Institute of the Philippines, as well as the Balik Scientist Program. Please watch this.
We are now living in a new normal. This COVID-19 pandemic marks as one of the global challenges experienced in this generation. It forces every sector of our society to innovate in order to move forward. We, at the Industrial Technology Development Institute of the Department of Science and Technology, is trying every possible ways to continue our service to our people, without compromising the safety of each and everyone.
tracing its roots back in 1901 from the then Bureau of Government Laboratories, which became the Bureau of Science. The Department of Science and Technology Development Institute, or DOST ITDI, turned 119 years old on July 1, 2020. From basic researches to mapping of the country's flora and fauna and other local resources for scientific studies, DOST ITDI has been a vital instrument in establishing the research and development agenda in the country. It was 1958 when the Bureau of Science became the National Institute of Science and Technology, NIST, that industrial R&D started gaining ground while harnessing local resources and skills towards self-sufficiency and optimized productivity. Industrial R&D went full gear in 1987 when the NIST was renamed Industrial Technology Development Institute, focusing on 4 major functions: research and development, technical services, technology transfer, and custodian of the national units of measure to provide international traceability. DOST ITDI Research and Development covers 5 major areas: food, environment and biotechnology, chemicals and energy, material science, and packaging technology.
The DOST program on the establishment of Virology and Vaccine Institute of the Philippines, or Balik, was conceptualized to address the pandemic readiness of the country. The objectives of the program are: 1. Serve as the premier research and development institute in the field of virology, encompassing all areas in viruses and viral diseases in humans, plants, and animals. 2. Act as a venue for scientists, both here and abroad, to work collaboratively to study viruses of agricultural, industrial, and environmental. And 3. Establish strategic partnerships with the world's leading scientists, virology centers, and institutes, and conduct innovative and pioneering research that will advance the frontiers of virology in the country.
Recognizing the critical role of science and technology in economic development and progress, the Balik Scientist Act, or Republic Act 11035, was signed into law last June 2018 by President Rodrigo Roa Duterte.
This is actually putting into law a program that has been started by the Department of Science and Technology almost 40 years ago. But we need some legal support so that we can implement it in a better way. The Balik scientists will have easier time in terms of coming here to the Philippines and rendering services.
Who is the Balik scientist?
Experience and expertise of Filipinos who have made good practice of being scientists abroad, so that they can share whatever they have in terms of knowledge and wisdom, institutions, our own researchers.
My role and responsibility of a Balik scientist is to be not just a teacher or facilitator, but also a pusher of innovations within the program.
Balik scientists are given support by the government for their stay in the country and are likewise provided with a wide array of benefits to ensure their maximum output.
The best incentives or privileges or benefits are having to be exposed with our farmers.
My greatest privilege would be doing collaborative work with fellow Filipinos.
The DOST, as the leading agency in charge of the Balik Scientist Program, is tasked to facilitate the placement of the Balik Scientist among its priority areas from its sub-agencies. P-Card, PCHRD, P-Shared, Partnering with the DOST are the host institutions, private or public entities, providing the appropriate resources to the Balik scientist in the completion of their research activities and other tasks.
I think the role of the institution is to give the space or the laboratory needed for the program or the project.
Working together, the DOST, the Balik scientists, and the host institutions have proven the importance of collaboration and cooperation, critical of any nation's vision for success.
I am an advocate of Balik Scientist Program. Okay, being one, I really truly felt that Balik Scientists would be able to help in uplifting the economic growth of the country.
My hopes is to really contribute to the space agency. It's very, very pragmatic, and we will need everybody's help, and also promulgate the STEM program here in the Philippines.
We really need to encourage other Balik scientists or other scientists abroad to give their time. They need to give back and help the country.
With the enactment of the Balik Scientist Act, the country is looking towards a stronger and more solid science and technology foundation, propelling the nation to further heights. Change has come indeed for science and technology. Science for change. Science for the people.
Thank you very much for that audiovisual presentation. This time, I would like to acknowledge the presence of the Director of ITDI, Dr. Annabel V. Briones. So, just a reminder for our Zoom participants, kindly rename your Zoom account to this format, your affiliation _ your full name. Like for example, in my case, DOST-ITDI _ Joseph Ancla. Do not also forget to mute your microphones to avoid interruptions during the presentation proper. Also, please answer our webinar evaluation form at the end of the lecture to secure a copy of your certificate. If you have any questions or clarifications for our speaker in this webinar, you may key it in our chat box in Zoom. Our audiences from our Facebook and YouTube live stream may also type in their questions in the comment section so that our moderators can pick them up and be answered by Dr. Jasmilio. For this second day of the webinar, we shall still be learning a lot about the basic concepts in virology, the virus replication cycle, as well as the infection and transmission process from our resource speaker, Dr. Myra Hosmilio, a Balik scientist from the Division of Virology, University of Cambridge, United Kingdom. But before I turn you over to our speaker, let me give a recap of our session last Wednesday. First of all, Dr. Myra discussed the viruses of clinical and economic importance in the Philippines, which include SARS-CoV-2, dengue virus, HIV, rabies, African swine fever virus, measles, polio, and Ebola reston. She also talked about the existence of viruses in our daily lives, wherein viruses infect all living things such as human, bacteria, and phytoplanktons. We also regularly eat and breathe billions of virus particles, and we carry viral genomes as part of our own genetic material. A very good example of this is the LTRs, or the long terminal repeats. We also have short and long interspersed nuclear elements. And in spite the vast majority of viruses that infect us, they have little or no impact on our health or well-being because we have an amazing immune system, like the innate and adaptive immune response. She also defined a virus as an infectious, obligate intracellular parasite comprising a genetic material, so it can be DNA or RNA, and often surrounded by a protein coat called capsid, and sometimes a membrane. In addition, she tackled the different properties of a virus, starting with their size. So viruses are very small, ranging from 20 to 700 nanometers. Again, they are obligate intracellular parasite. So they just float around in the environment until they come in contact with the appropriate host. So they are also composed of nucleic acid and proteins, sometimes with lipids and carbohydrates too. They also have a unique mode of replication process, and this will be discussed this afternoon by our resource speaker. When it comes to diversity, all species that are infected by viruses may cause great plagues or be asymptomatic. She also mentioned a short history of viruses, wherein people thought at first that viruses are in liquid form until the discovery of the electron microscope in 1930s. Furthermore, she discussed the principles and functions of virus structure. Virus structure is not a very simple process and usually involves capsid symmetry, axes, vertices, and faces. Virus structure can be studied using electron microscopy, X-ray crystallography, cryo-electron microscopy, and nuclear magnetic resonance, or NMR, to understand the specific component of proteins and their symmetry. On the other hand, viruses are composed of non-structural and structural proteins, and the main function of these structural proteins is to protect and facilitate the delivery of the viral genome to the host cell. Lastly, according to Dr. Hosmilio, viruses can be classified according to the nature and sequence of nucleic acid in virus particle. They can also be classified according to the symmetry of protein shell or the capsid. So we have icosahedral, helical, or complex viruses. Another classification would be the presence and absence of lipid membrane. That's why we have viruses, we have envelope and non-envelope viruses. So viruses can also be classified according to the dimension of virus particles. So these are the recaps of the first part of this webinar. For the second part, let us all welcome again Dr. Myra Hosmilio to discuss the virus replication cycle and the infection types. A virtual round of applause for our speaker. Dr. Myra, the virtual floor is now yours.
Hi there. Good afternoon. Really, thank you, Joseph, for that. It was a very good recap, although I also prepared a few slides for my recap, but thank you for that. I'll stop my video and then just share my screen for now. I thought I'll use the laser pointer this time. Anyway, so isang magandang hapon sa inyong lahat and isang ulit na magandang umaga mula dito sa Scotland, okay? So as a virologist, one of the important things that is very important, one of the important things that you really want to have when you want to explore a virus virology and learn more about viral pathogenesis is to be able to dissect the virus infectious cycle. And so to have a good, robust cell culture system is very, very important. I have here a photo of one of my projects when I was in Korea. And this was, at that time, I was studying porcine sapoviruses. Porcine sapovirus is a good carrier of, it's a virus that causes diarrhea in pigs. But we know that another member that shares the same family with sapovirus, which is norovirus, also causes diarrhea in humans. So having porcine as a model would be good to understand human noroviruses. But at that time, I was really struggling to grow the virus. And one of the techniques that I have developed and other people in Cambridge has helped me, and I have been using up to now whilst I study SARS, is to engineer the cells in such a way that here, I engineer cells in such a way that they express viral proteins that I know targets innate immune response. So it's a bovine viral disease virus, and an N protease specifically targets IR3, and this targets interferon production, while PIV5V targets interferon signaling. So as you can see, and this is a plaque assay, that without inhibition of the innate immune response, my plaque assays are quite small, and with the inhibitors of innate immune response, the plaque assays are significantly bigger. And why is this important? At that time, I want to explore how viruses hijack the host machinery. I wanted to understand the innate immune response in cells due to virus infection. I want to identify small molecule inhibitors that can be useful to prevent virus infection. And it only will allow you if you have a robust cell culture system, because that will allow you to get more viral RNA, more viral proteins to study further, okay? So today, the aims of my lecture is really to give a quick recap. I know Joseph has done this quite well. I'll just give the others that he has not mentioned so that we can move through and I can discuss more thoroughly on replication cycle and virus basics and virus infection. Because we have time, because I have restrictions with time, I have actually prepared slides about going through general types of infection as well as transmission, but I'm hoping you will just read this through by yourselves, and you can always email me just in case you've got questions. But today I'll concentrate more on replication and basics of virus infection. So let us begin. So what are the viruses? What is a virus? So the 3, and 4. These are the 4 main characteristics of the virus. They should be infectious, they are infectious. They're obligate intracellular parasite comprising genetic material, which can either be a DNA or an RNA, and they can be surrounded by a protein or sometimes a membrane. So these are the important viruses in the Philippines. This is SARS-CoV-2, this is HIV, both of these are RNA viruses. This is, I think this is dengue virus. This is ASFV, and ASFV is a double-stranded DNA. These are negative strand RNA viruses. This is rabies, and this is measles or morbillivirus. And the largest one, which is a helical in shape, is Ebola virus. But we have Ebola reston in the Philippines. Okay, for virus classification. So right now, the classification that we follow for viruses is a Linnaean hierarchical classification, where we classify virus according to family, genus, and species. But I just want to mention very briefly that in the past, it's not the case. As I have said yesterday, the viruses can be named according to associated diseases, such as polio and rabies, the type of disease that it caused, such as murine leukemia virus, the sites in the body affected, such as rhinoviruses or adenovirus. Some viruses are even named according to the places where they were first discovered, such as Sendai and Coxsackie virus. Scientists who discovered this virus and Epstein-Barr virus, and even the way people imagine these viruses, such as dengue virus, which actually means evil spirit, or an influenza, which actually means an influence of bad air. So these are just a very personal way of, or subjective way of classifying virus. So if you remember very recently when Wuhan virus or when SARS-CoV-2 was pertained to be Wuhan virus, that was not nice for Chinese, because it can be associated that the SARS-CoV-2 and all the bad things that happened of SARS-CoV-2 was just due to China. So we try to avoid that, and that's why we have a more objective way of classifying viruses. So I just want you to remember as well that in the classification of viruses, they are grouped according to their properties and not according to the cells that they infect, okay? So because of the development of genomics, we can easily classify viruses based on the sequence of the nucleic acid. But there are actually 4 characteristics, and Joseph has mentioned that. I am just attaching here the organization that deals with taxonomy of viruses, and you can easily Google this. It's an International Committee on Taxonomy of Viruses, or ICTV. So every year it gets updated. So for example, when I was studying hepatitis E virus, it was first classified as calicivirus because the virus was identified by EM and it was thought to be similar to caliciviruses. However, when they learned more about the sequence of the virus, they moved that to herpes viridae. Okay, so it can change based on the knowledge that we gain. And I'd like you to memorize this another way of classifying viruses, because this would be necessary for better understanding the ways or the strategies that how viruses replicate in cells. This is called the Baltimore classification, and it's classified according to the nature of the nucleic acid, which, as you can see here, they're classified until 7. They're positive sense RNA, negative sense RNA, DNA, single-stranded, double-stranded, so these things. And this tells you how viruses enter into the cell and propagate. So one question that I have received yesterday was about 2019 SARS-CoV, whether it has already been classified. So it belongs to order Neuroviralis, suborder Cornidoviridae, family Coronaviridae. So coronavirus is a huge family of viruses. And so SARS-CoV-2 belongs to Orthocoronaviridae, genus Beta coronaviruses. So the ones that cause common colds are usually alpha coronaviruses. Subgenus Sarbecovirus, and the species is severe acute respiratory syndrome related coronaviruses. So here is SARS-CoV-2, Wuhan, and it is classified with the SARS-CoV-1, the civet SARS-CoV-1, and bat coronavirus and coronavirus from rat, because genetically or by genetic sequencing, they are the closest. And you can find this more in this paper. So the process, as Joseph said, of characterizing a virus structure is definitely not easy. And more importantly, it's extremely expensive, okay? A long time ago, we just have EM, and that is totally enough. And it's really allowed to be able to identify a virus by EM. However, there are limitations with EM because the resolution is only until 50 to 75 Armstrong. And by EM, you will not really be able to learn more about physiochemical characteristic and do a 3D manipulation. But nowadays, we have better or advanced technologies such as X-ray crystallography, and with X-ray, viral proteins has to be crystallized. And by principle, the X-ray hit these crystals and it forms diffraction patterns, which is collected here. And the diffraction patterns tells us about the structure of a viral protein. Nowadays, most scientists use cryo-EM, another very difficult and very expensive process. In cryo-EM, it depends on freezing, and then that's why it's cryo. Cryo means like frozen. And then an aqueous solution is added, and the change, in addition, change the solution, and there's a lot of computations gained or collected. So when you freeze a viral particle, they are frozen in different forms or orientation. And these are all collected by computers, not just hundreds of images, like really thousands of images. And from that collected information, you can form a 3D reconstruction. Another question that I got last time, where I want to correct myself, is on NMR. When I was a student, what we usually do is to make sure the protein is crystallized, and we send that for NMR. But I've read through more on NMR, and actually, NMR is particularly important when you're studying smaller proteins, okay? When you say small, that usually are 15 kilodaltons and a lot lower, okay? But the process requires your protein sample, your viral protein sample, to be exposed in a magnetic field. And when you do this, it has to be combined with a deuterated solvent. So it has to have a solvent. So it is actually a solution, but it should not, there's only a limited volume of solvent that you have to add, so it's not to dilute. You also don't add water because it will degrade the protein. You specifically use a deuterated solvent because that does not affect the magnetic field around. So when it's exposed, it produces like electron waves, and that is called a precision. And this is measured, and all this calculated precision tells about the structure of a viral protein. Okay. So the structures according to symmetry, I just wanted to give a recap on this, because whenever I myself read 3D structures or a structural based paper, it can be difficult to understand. But you have to understand that in a protein, they produce a subunit of a capsid. So usually, for example, in SARS-CoV-2, they have at least 4 subunits of the structural protein, okay? But sometimes a protein can have just 1 subunit, and then 1 subunit is repeated and forms a symmetric orientation. So as you can see here, there's 1, 2, 3 subunits of the virus that forms a helical symmetry. This is one subunit of a viral capsid, which forms an icosahedral symmetry. This is a protein with, so you can see there are different colors. So 1, 2, possibly 2 subunits of virus capsid that is also forming icosahedral. So is we automatically think that round viruses forms an icosahedral symmetry, while rod viruses forms a helical symmetry, and all the others are classified as complex. There are specific numbers to form this symmetry. This is another term for symmetry of viruses that exceeds the required amount for icosahedral, and it's called quasi-equivalence. So as I have said, we've been talking about viral structural proteins, but because we're studying and entering, and now we'll learn to understand virus replication, I'd like to introduce the other components of virus particle. And so you can see here, there's actually a lot of like structural proteins, but inside there are proteases, there's a viral RNA. These are composed of enzymes, polymerases that are important for RNA synthesis, proteases, also important for replication. There are even activators, mRNA, for activators for mRNA degradation, and on some other cellular components. So some of you might be very familiar on this because we've been always seeing the SARS-CoV-2 genomic organization. So this is how it's laid out. It is composed of at least 14 proteins. So this part is the structural protein of the virus. You can see, and the main subunits are spike, EM, and nucleocapsid. ORF1 and ORF1, it should be ORF1A and ORF1B, comprise the non-structural components of the virus. And actually, if you look at this paper, all of these non-structural proteins that range from non-structural protein 1 or NSP or NS1 to NS16, each of these proteins have a particular function for virus replication. For example, NSP1's role is to inhibit the host translation so that it can allow virus protein synthesis. Of course, there is RDRP, which is very important for virus replication. NS4 is important for modifying the virus when it's in ER. So there are really several functions, and they are all advantageous to the virus. So today, we'll concentrate on virus replication, Virus replication can also be called reproduction cycle. And the infectious cycle of a virus means that it comprises every step that happens in a virus from its entry to virus release. Okay. Virologists or people who study on viruses tend to divide them in several steps for us to easily study, and also because in several steps we will be able to exploit these steps so that to identify specific small molecule inhibitors or antivirals against the virus. So this process here is specific for SARS-CoV or SARS coronaviruses. The virus attached to its receptor, which is ACE2. TMPRS is a host protein which facilitates virus fusion, and the virus is endocytosed. Now it's in an endosomal vesicle. There are changes in endosomal vesicle. It could be a reduction in pH, which allows to release the viral RNA. Because the viral RNA is a positive strand RNA, it immediately serves as the mRNA, which allows the immediate translation of viral proteins. SARS coronavirus is a particularly large virus because it has 30 kilo bases. And so it forms 14 open reading frames, which corresponds to both structural and non-structural proteins. So as I've said, immediately the virus used the host machinery to make its protein. It comes as polyproteins first. These are immature proteins, which is then processed by viral proteinases to have mature proteins. These mature proteins, they are important to have a replication complex, which allows replication or RNA synthesis, synthesis of positive RNA synthesis of negative RNA, subgenomic RNAs. So once they are produced, as well as the proteins are produced, the virus starts to do its assembly. So as you can see here, the structural proteins are formed, and it engulfs or assembles the non-structural proteins. And after the virus assembly, it goes out of the cell using the host cell exocytosis machinery, and it leaves the virus. So they're just very important. There are important definitions that I would like you to remember again. So one of those is susceptible. So susceptible is a cell that has a functional receptor. Okay, as you can see, as a functional receptor for a given virus, it may or may not be able to support viral replication. As you can see here, this is the virus, there is a functional receptor. And because of the functional receptor, it is able to support virus replication. Here, I think there is no functional receptor. A resistant cell is a cell without any receptor. So these may or may not be competent to support viral replication. So there is no receptor here. So, but there is virus infection, and that did not happen naturally. Usually the presence of an infection without a receptor only happens when you introduce the viral RNA directly inside the cell. And this is done through a process called transfection. The other picture here shows no virus replication at all because there's no receptor. Permissive or permissibility. A permissive cell has the capacity to replicate the virus. So the virus is able to replicate inside the cell. As I have said, without a receptor, the virus can be permissible, but that only happens when the viral RNA, the infectious viral RNA, is introduced into the cell via a receptor, via transfection. So the two main important definitions that I would like you to remember is actually susceptible and permissive. So the susceptible and permissive cell is the only cell that can take up virus particle and replicate it. Let's go to culturing viruses. So for many, many years, scientists don't really know how to culture viruses. And so long time ago, what would happen is that they would use animals to do the experiment and to produce viruses. For a very good example is with polioviruses. A long time ago, scientists would use non-human primate to infect poliovirus, and they would collect the brain and the spinal cord and grind them and then collect the virus from them. During those times, there was also no freezer. So you could imagine for somebody to produce a vaccine for poliovirus, an inactivated vaccine, they have to do this process again and again and again, and they have to kill a lot of animals. And importantly, they may also be using infecting animals, infecting the wrong animals, although it produces viruses. If it's a wrong host, then it can lead to a selection for variants. But very recently, not recently, but from 1949, we were able or scientists were able to start growing the virus. And that is using an embryonated chicken eggs, which is about 12 to 13 days fertilization. So here, as you can see, the virus can be inoculated in different parts just because viruses targets different cell types. And in Korea, what we did before was when we were trying to check whether there is influenza in farms, we would collect stool samples and inoculate the filtrate into the allantoic here, which is quite easy because the needle penetrates their sac, and then you inoculate the virus here. And then after a few days, we collect the viruses. And a very good implication of this is in terms of growing flu vaccines. To be able to have flu vaccines, we actually use inoculated eggs. And we use 150 to 200 millions of eggs to be able to produce flu vaccines. There are different methods to grow the virus, just in case we have a pandemic. But I think generally, until now, we've still been producing flu vaccines using egg inoculation. So this is just an automated way of inoculating viruses and egg. You can also do it yourself. As you do repetitively, you'll be good at it. And these are just some of the viruses that are specifically inoculated at certain positions of the egg. So the cell culture system also came just around 1950. And the scientists who discovered this received the Nobel Prize. And from then on, we have been using cell culture for growing viruses. This is a primary cell culture system, which uses a human foreskin fibroblast. The primary culture system can be very good because they can be more closer to more natural viral processes, not exactly, but a little bit better than immortalized cell lines. These are immortal or cancer cell lines, which can grow forever. And one of the very famous cell lines are the 293 T cells or the HeLa cells, which you can grow in the lab, you can stop growing them and stock them in liquid nitrogen, and you can revive again out of the liquid nitrogen, and you can just go on and on and on and on and on. Just the problem in using immortalized cell line nowadays is they may or may not have the right machinery inside the cell culture. So it's usually not used to a good system when you're trying to grow vaccines, because it is possible that it can have an effect to the end users. When nowadays, there are also amazing advances in the cell culture, and I have been using this in the lab. So as I have said, there are limitations of using immortalizers, but they have been really useful. However, nowadays we try to find cells that are more physiologically and pathophysiologically similar to the target hosts of the viruses. So for example, in the lab, I have been growing a human norovirus, and the system we use is pediatricians collect biopsy sample from patients. From those biopsy samples of intestines, we isolate the stem cells, and from the stem cells, we grow intestinal organoids. And it's quite a good system because it doesn't only contain one cell type, it contains 4 different cell types, which is just as similar to a real intestine. And it also has villi, it also has crypts. And for viral studies, for example, with rotaviruses, you can see swelling and you can understand the pathogenesis of diarrhea by using intestinal organoids. We've got a collaboration with a company in America where they based their preliminary tests on humans from our experiments, because and their findings is also quite similar, because we tested their drugs in intestinal organoids. What they see in human is quite similar. Nowadays, you can make organoids from almost every different parts of your body. So there are brain organoids that we can use to study Zika. There are also organoids of the ear, organoids of the eye, name it. You can have it nowadays. And that's because of the power of stem cells. Very recently, I have done experiments in the lab with coronavirus using an air-liquid interface culture system. And this is quite interesting because, especially for when you're studying respiratory viruses, this is usually set up in a trans well. On top, there is no media, so it's just literally dependent on air. And then at the bottom, there is a liquid phase. So, and these cells can be mucus producing, could be ciliated cells. And this type of plate is actually called transwells. And yes, I have done this experiment. It's quite amazing because you'll see movements of cilia even in the light microscope. And once you add the virus, the cilia can go crazy. But it is interesting. So this system, for example, on the normal cancer cells are just plated on plastic plates without any support. Organoid culture system has to be suspended in a gel form, what we call is matrigel. But in the case of an air-liquid interface culture, it has to be in a transwell. So once you infect the virus, what we try to observe is the presence of a cytopathic effect, or what we call CPE. These are just some of my experiments that I did. So this is just inoculating an intestinal organoids with an Indian strain of the SARS-CoV-2. You can see that without the virus, there is no CPE. With the virus, there's very clear CPE. In the lab, we also engineer the virus such that it produces a green signal, or GFP fluorescence. And in this way, we are able to study protection assays and neutralization assays using serum patients, serum of patients. So CPE comes in several forms, such as usually we look for rounding of cells and detachment of the cells on a plate. But in particular with coronaviruses and some paramyxoviruses, they form a syncytium or cell fusion. And that is when single cells fused with 2 or more cells. Okay. Now we go to, I just want to say that the CPE is usually observed depending on the virus. Usually when we grow viruses in the lab, we observe the viruses that we grow 48 hours post-infection. And this leads us to the definition of virus growth curves. So to understand virus reproduction in cells, we should know a virus growth curve. And this was also developed by virologists, at least in 1940s. And as you can see here, these plots are plotted in such a way that y-axis represents the infectious particles. It could be a PFU, it could be a TCID50, against time on the y-axis. Here you see, and I have discussed this, that in viruses, in the virus growth curve, for the first, in its initial period, you'll not observe virus particle, and this is called the eclipse period. And once the virus particles are made, there is a yield or burst period. And this can vary depending on the amount of virus that you introduce into the cell. So this is called a single or one-step virus growth curve, and this is a multiple growth curves. When you inoculate a cell with a lot of virus particles, then you expect that all cells get infected and they all behave in a similar way, and such so that you can see a very clean time point for when you'll see when you see the virus particles. However, it is possible that you also manipulate the virus inoculation in such as you just inoculate lower titer of the virus. And in that way, you'll see an initial burst, and then it will be followed up by a secondary burst or a third burst, or so on and so forth. In the case of adenoviruses, an adenovirus is usually a virus that causes respiratory and enteric problems in animals. And adenovirus is also a vector that is being used for SARS-CoV-2 vaccine. So its virus curve has an eclipse period and the burst period. However, this graph is plotted in a little bit different manner because there is an intercellular component and extracellular component. So when you infect your cells, you can actually just collect the supernatant, and we see as the released virus. And you'll see that in cells, you can already detect virus particles 12 hours post-infection. However, in a supernatant, or in the virus released, is only detected 16 hours post-infection. And that period is called the latent period. So I have been talking about the growth curve, and I've said about the single growth cycle and then a multiple. And this leads us to the definition of multiplicity of infection. So you can manipulate, when you are studying viruses, you can manipulate the amount of virus that you can add. And this is called, we always say, what is your MOI? And that means the number of infectious particle, virus particle that is added per cell. And this is not a simple mathematics because it follows a Poisson distribution, which is this equation. And so it is not exactly the number, not the number of infectious particle each cell receives. So when you say, and that means that when I put 1 million cells, that means each cell will receive, when I put 1 MOI, each cell will receive 1 virus particle. It's not the case because of it happens in a random event. And through this Poisson distribution, we know that when we put 1 MOI of virus inoculation, you'll infect 63% of the cells. When we inoculate with 8 MOI of PFU per cell, then we can achieve 100% of infection. Okay. So, are you still with me? I have a question. So A blank and blank cells is the only cell that can take up virus particle and replicate it. What is the answer? And the answer is a susceptible and permissive cell, okay? So the next part of this lecture is on the step-by-step of virus replication. So the first step of virus replication is virus binding. And during virus binding, the host molecule that is used for the attachment of virus is called the receptor. The viral protein that attaches to the receptor is called the viral attachment protein. Okay. In the case of SARS-CoV-2, the receptor is ACE2, and it's called the epitope that binds to ACE2 is called receptor binding protein. And studying this is quite important because the expression of receptor can determine the virus host range and tropism. Yesterday, somebody asked about mixing vessel. In the case of the poultry, chicken influenza or avian influenza, they usually target an alpha-2-3, and a porcine influenza targets alpha-2-6. So humans don't have alpha-2-3, so it's really impossible for humans to have avian influenza directly coming from chicken. It has to have an intermediate host. And we think that also happens for SARS-CoV-2. It is important to say that receptors have not evolved to allow viruses to enter cells because they have their own functions, okay? So as I have said, it was very, very quick when scientists have identified H2 as a receptor. One reason is because SARS-CoV-1 utilizes ACE2. And this is just an experiment where the scientists use HeLa cells. HeLa cells don't normally express ACE2, or if they do, maybe a very, very low level. When they express human ACE2, you'll see here, this is an ACE2 that is marked by a GFP. And this is a nuclear protein of SARS-CoV-2, which is using an antibody against nuclear protein. And you can see that there is the virus when you've got the human ACE2. The virus is also there when you've got the bat ACE2, but it's not there when you have the mouse ACE2. And this is really very important and very, very interesting information for us to be able to understand the virus, for us to be able to design drugs against the virus. In the cells, or the cell has a bilipid layer. But within that bilipid layer, it is composed, or it consists a bunch of proteins, okay? And there's several of them. And the virus uses different proteins for its entry. There are even components of the phospholipid layer, which can also be utilized by viruses for its entry. And these are just examples of cellular receptor that is being utilized by particular viruses. As you can see, some viruses even use more than one receptor. Viruses that belong to similar family may or may not have similar cellular receptors as well. So they vary, and these are always part of the investigation. Once the virus binds to its receptor, it then goes inside into the cell through several pathways. And this is just a normal way of entering different macromolecules or molecules, as a normal process for cell. They can go via phagocytosis, endocytosis, such as iron, phagocytosis for bacteria in macrophages, and a filopodial capture. But in particular, the virus utilizes the endocytosis mechanism. It's either by macropinocytosis, some viruses use it, but most of the viruses use a receptor-mediated endocytosis. I'm seeing the time now, so I will just be a little bit quicker, okay? And just give me 5 minutes to finish what I can do. In an endocytosis, viruses uses different pathways. First of all, here you see that the virus fused with the plasma membrane, and it enters into the cell, and it gets transported immediately into the nucleus via these microtubules. And it's quite common with adenoviruses. This is another virus, which is this time is endocytosed and is formed in an endosomal vesicle. And this is also carried along in the cytoplasm. Other viruses use different types of endocytosis, which involve clathrin-dependent, clathrin-independent, caveole-independent. So these are just different ways to enter or to enter a cell. So this is a ribbon structure of the SARS-CoV-2. This is the spike protein. This is the receptor binding protein. This is the ACE2. So in its spike here in cyan is actually the fusion protein, but it is not yet exposed when the virus is not yet attached into the cells. Once it's attached, it gets cleaved by cellular proteases. One is TMPRS. And there's another area here which is called the furin cleavage site, which is also cleaved by furins and host furins. And upon attachment, these fusion proteins are exposed, and it allows fusions into the plasma membrane so that it gets endocytosed. And this is quite cool, right? Viral replication strategies. So there's several viral replication strategies according to the nature of the nucleic acid. So, for example, I have said a while ago that once a positive RNA virus enters, it immediately produces its viral proteins. But it must compete, and it usually competes with the host mRNA. And so there are different viral strategies that allows protein synthesis. For example, there are viruses that has IRES. These are highly complicated structures where the host translation machinery gets lodged in here, and it allows virus translation. For other viruses, such as caliciviruses and poliviruses, picornaviruses, they can utilize, or only caliciviruses, they utilize VPG. It's a virus-encoded proteins that allows viral protein synthesis. So in the case of the virus, SARS coronavirus life cycle, so there is binding, endocytosis, production of proteins. SARS coronavirus translation is cap dependent. That means it uses a similar translation mechanism as the host. That's why it should compete. That's why you know that one of its proteins' role is to inhibit host translation. So it allows virus translation. And one of the proteins that is produced is polymerase. The polymerase is used for RNA synthesis. And that's positive, negative subgenomic RNAs, which is then used for assembly and egress or egress. For viruses such as negative RNA genomes, they have a different way. Every virus must have mRNA to produce its viral proteins. But for them to convert itself from negative strand RNA or DNA to become mRNA, this is the basic requirements. So most of the virus, for example, negative RNA genomes already have RDRP along with the viral particle. And so once the virus is assembled, it's released out of the cell, and this happens in several ways. But also I want to stress that this uses the host exocytosis machinery. So all viruses just try to utilize the machinery and try to compete with the cell to allow specific viral processes. There are several ways to confirm or quantify virus. You'll hear about PCR, plaque assay, TCID50, and foci forming units. I'll leave this to whoever want to ask question if you want the detail, okay? And so, question: virus receptor on the cell surface can, all of the above, bind directly, interact with glycoproteins of envelope viruses, can be carbohydrate protein molecules, and all of the cell surface receptors actually have their own cellular functions. Positive RNA strands are immediately translated, yes, and the process of exiting the cell is egress, but not endocytosis. It should have been exocytosis. can I just ask to my, can I just ask to if I am still able to continue or we should stop and we proceed to question and answer. I'm just conscious about the time. Joseph, can you please advise if I can continue or we stop and we
Yes,
go for
ma'am, you may
question?
continue pa din po. We still have a little time.
Okay, thank you.
Thank you.
So I would like to go through very quickly on the basics of virus infection. To be honest, when I was trying to prepare my own this presentation for you, my goal is to give you as much knowledge as I can, and then my intention is really to give all the knowledge that I have. But we just have restrictions and limitation. I hope I still have another maybe chance to go through, but you can always ask questions, okay? So one of the important terminology or parameters that we use during virus infection is incubation period. Incubation period is the initial period before symptoms of the disease are obvious. And the signs that are present during incubation period, or what happens is that viral genomes are replicating, and whilst they're replicating, the host cells are responding. So they're all innate immune response getting triggered, and stimulated. However, during this period, the virus may or may not be transmitted during an incubation period, okay? A very good example is Ebola virus. During incubation period, Ebola virus is not transmitted. So people would fly, and whoever they're side by side, they will not get Ebola virus. The virus is only transmitted upon onset of the disease. However, this is very different with SARS-CoV-2, because with SARS-CoV-2, whilst we are in an incubation period, we are already transmitting the virus. And so whoever we're sat with, or one cough, and then another complicated thing with SARS is there are also symptomatic patients who can transfer and easily transfer the virus, okay? So typically, a virus with short incubation period reflects that the replication is at the primary site. So for example, in the case of SARS, the virus is in air, we inhale those, and the virus hit your epithelial cell, and then your linings, they may have ACE2, and then they can start virus replication. For other viruses, which is long incubation, usually the symptoms are not in the primary site, but in the secondary site. So for example, in the case of rabies, you get infected by an open wound with saliva. They go into your blood and into the brain, and then there is the onset of the disease. So yeah, that's why when somebody gets bitten by dogs, they have to have vaccination immediately. And sometimes with rabies, the onset of the disease also come in years. So yes. Different types of infection comes with either acute, persistent, and latent. Acute is a rapid onset of the disease. There are several acute viruses that includes a lot of respiratory diseases. Not all acute viruses causes disease. The body is able to resolve them, so you do not necessarily get sick. But in the case of Ebola virus, once there is an acute infection, but the degree of mortality or case fatality is really very high. Persistent is when a virus still remains in the specific cell type that it is infected. And most of the persistent virus usually have a ability to control the innate immune response. And so that allows them to not be resolved out of the body, and they continue to replicate. The last one is a latent virus. These viruses usually stop from growing, but they are not eradicated, just the proliferation stop. And this happens usually during situations where the innate immune response is depressed. They sort of tend to be like opportunistic, and then the virus particle increases. So there are different notes on this, such as morbidity, mortality, incidence, and fatality rate, which is quite important when you're trying to understand virus infection. So incidence is the number of people infected over the population, and it can be measured in different ways, in different time points, like per year, per month, 7-day period. So here, the incidence is 25% because there are 5 people infected within 25 population. Morbidity is the number of people ill that got ill in the population. So you see, again, there is 2 people. These are having signs and symptoms within the 25 number of individuals. So this morbidity is when they have shown the signs and they're still positive of the virus. Mortality is the death in the population. So that is there over the 25, hence it's become 5%. And we can calculate case fatality rate, which is the number of deaths over confirmed infected. So this can be compromised because, for example, with SARS, not everybody confirms the infection. So sometimes we expect that the case fatality is given quite higher, but actually the number of confirmed infected could have been higher, okay? And the infection fatality rate is the number of deaths based on actual infection. And the last one is quite difficult to know accurately. So one has to extrapolate and do statistical measurements. Now let's go to a basic reproductive number, or R naught. This is just the number of secondary infections that can arise in the population, okay, of susceptible. So you already know from the media and from every information around that when the R naught is greater than or less than 1, an epidemic cannot be sustained. When an R naught is greater than 1, an epidemic is possible. But much greater, the epidemic is certain. In the case of SARS-CoV-2, in the beginning of the outbreak, we know that it has an R naught of 2 to 3. And so that's why we expect that there will be increased outbreaks at certain period of time. But the R naught is influenced by time, influenced by several interventions, and that includes lockdowns, and it includes vaccination. So this is just a different way to look at case fatality rate in R naught. And for example, with measles, there is one person can actually infect about 15 individuals. However, the case fatality of measles is not as high as SARS. In the case of common cold, one individual can infect 1 or 2 individuals as well, but you don't really get sick. However, in SARS, it's quite varied, and I'll explain you very quickly. It's because the case fatality rate, although in general or overall, it says to be 2.3%, actually, if we stratify the infected individuals according to age, you'll see that younger individuals are not really getting really sick due to SARS-CoV-2. However, individuals which are very old and maybe having predisposing factor are the ones that have very high case fatality rate. So the case fatality rate really vary. Another important parameter is over-dispersion. When we say that the R naught for SARS-CoV-2 is 2 to 3, we expect an immediate increase in infection in the population. However, it did not really happen immediately, and that is because of dispersion. So for SARS-CoV-2, there's a K of 0.1, which means that of the 80% of transmissions, only 10% are infectious individuals. So only 10% are causing infections to other people. So it's not directly or immediately causing immediate infection, except for super spreading events. And this is a very, very good study in Hong Kong where they have identified super spreading environment. So it's not necessary that a lot of individual infects a lot of people, but actually one individual can infect a lot of people in a super spreading environment. And that includes wedding, that includes masses. And that's the reason why we had to have the lockdown. You see here, there are short chain events, but the massive ones that increases virus infection is the super spreading events, okay? So with that, and this is the last part of my slide, is when you have an MOI, what is the characteristic of growth curve that you'll see? Well, when you have an MOI of 10, you expect over 100% of infection. That means you should only expect a single burst of virus release. And then, which of the following factors is not influenced by human interventions? I think all of these are actually influenced by human interventions. The incubation period can be influenced by vaccination and all the other things. Okay. So with that, I would like to thank you for listening and for staying with me and being very patient. I am happy to receive questions.
Thank you very much for that very educational presentation, Dr. Rosmilio. So that ends our learning session for the basic concepts in virology. At this point, we are open for your questions, and I know that our speaker will do her best to answer the questions. Again, if you have any questions or clarifications, you may key it in our Zoom chat box, and you may also type in your queries in the comment section in our Facebook and YouTube live stream. So let me first start with the questions from our participants last Wednesday, Dr. Rosmilio. The first one would be, what are the common druggable targets in viral therapy? So this question is from Madison Munar in Facebook.
Druggable targets. There are several. So once we know the virus components of virus particle, particularly we always want to target the ones that is just present in the virus and not the ones that are being used by viruses that are part of the host cell. So we usually try to understand first the virus component. I'm saying this because for me, this is the spike proteins and this is the receptor binding domain. Okay, so we usually try to inhibit this part because we know that when virus enters and it uses ACE to inhibit ACE2 receptor. When we do our virus, and this is a very good example for SARS, when we do our virus infection, we actually use a drug called Camostat, and that targets ACE2. So that inhibits or blocks the virus from binding into the host cell. When you're trying to study the virus processes, and that's what we do, you can identify important host proteins that are particularly needed by the virus for it to grow, and sometimes not really needed by cell or just needed for very minor functions. And you want to target those because, and they come in all different parts of the virus cycle, and you want to target those to stop virus infection. So right now for SARS-CoV-2, we try to target proteinases of the virus. You can also target that because the protease is important for the virus. Whatever component is very important for the virus in its replication cycle is the one that it can be druggable. And right now we use available drugs to first test these viruses, test these drugs against the virus.
Thank you very much, Dr. Rosmilio, for that answer. The next question is from Mariel Velarde Capapas. What misinformation from the media about COVID-19 would you like to correct or clarify? Because you said last Wednesday, and I quote, that one of your struggle during this pandemic is to fight misinformation about COVID-19. It's because the media played a major role in giving story in a very different angle, in such a way that the public will better understand. But unfortunately, that angle can be harmful as well.
Yeah, one of the very good example that I've given is when the media tells that a virus wants to be more transmissible, okay? So the virus does not want to be more transmissible, because when you do that, you make the virus an active agent, okay? And so people will get really scared that the virus wants to be transmissible. That means it can just infect anybody. But actually, it's a passive agent. The virus becomes more transmissible because of mutation. But equally, also the media also says the virus mutates. This is a very normal process for the viruses, especially for RNA viruses, to mutate again and again and again, and that's for their advantage. However, every time the virus mutates, it's not always advantageous to the virus. It's just very amazing with the SARS-CoV mutation has really led to it being more transmissible, but there's still a lot of things that we don't know. But in general, although viruses mutate, generally they don't really cause serious pathogenicity. So do not make viruses a human and understand the virus. Really, I think I like what you just what ITDI has shown, that once you know your enemy, and you better understand how to respond rationally. And I would like to hope that Filipinos know how to respond more rationally.
Okay. Thank you very much, Doctora. And I think this is the last question for today. How can basic reproductive number influence the vaccine strategy? This is from Tricia May Campit from CLSU via Zoom.
So the R0 is a measurement of the secondary infections. When we have interventions such as vaccination, we prevent. So when you have a infected individual, even though they are vaccinated, often they produce a less severe disease or less coughing. Maybe not. There's even it could even be asymptomatic. So there is less tendency for people to cough. And one of the very good way of really transmitting the virus is with coughing, and with a cough, which has the virus particles and an air. Other people who handle that. And so with reduction in the severity of the symptoms, then you reduce the transmissibility, and this reduces the reproductive index of the virus.
Thank you very much, Doctora. I think in the interest of time, we will be ending the Q&A session, and we will proceed to the presentation of the Certificate of Appreciation. And now, To present the Certificate of Appreciation to our speaker, may I call on Dr. Annabel V. Briones, the Director of the Industrial Technology Development Institute.
Thank you, Joseph. Can you please show the certificate appreciation? Doc Myra, thank you very much for your very informative lecture and also a crash review of the basic concept of virology. So, the Department of Science and Technology, Industrial Technology Development Institute awards this Certificate of Appreciation to Dr. Myra T. Husmilio, Balik scientist, University of Cambridge, for sharing her expertise as a resource person for the webinar entitled Basic Concepts in Virus Structure, Classification, Viral Replication and Infection as part of the establishment of the Virology and Vaccine Institute of the Philippines program of the Department of Science and Technology, held online on October 13 and 15, 2021 via Zoom, given this 15th day of October 2021, signed yours truly, Annabel V. Briones. Thank you very much, Doc Myra.
Thank you very much po. Thank you sa lahat ng makinig at thank you sa patience nyo dahil kahit na nag-extend ako ng time.
Yeah, so let's hear the participants here. We still have a series of webinars in the next coming months up to 2022. Joseph will inform it further. We'll discuss it further.
Thank you po, Dr. Annabel. Thank you very much to all who joined our learning session for today. Please answer this evaluation form flashed on your screens to secure a copy of your certificate. This has been Joseph Ancla of DOST ITDI. Do like and follow the Environment and Biotechnology Division ITDI Facebook page for announcements regarding future webinars. Maraming salamat po at magandang araw.
Thanks, Joseph. Thanks po, Mama Annabel.
Thank you po,
Thank you, Doc May.
Dr. May, salamat po.
And thank you to all participants. And to Joseph, thank you.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin, mahirap man ay kakayanin. Sa pinagsamang lakas at galing, tagumpay ay mararating. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya.
magkaroon ng cheap alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na